All right, we're going to move on to the second half of chapter 3 in week 1 and start talking about microscopy. So, obviously, we use microscopes to look at microorganisms because they can't be seen, most of them, with the naked eye. There are all kinds of different microscopes that can be used. They're specific to whatever type of microbe we're looking at. In this case, I've left some of the learning outcomes up. Sometimes I do, sometimes I don't. Sometimes I think it adds just too much text um, to go back and check about to to review what we're looking at. Sometimes I think it's helpful. So some of the things we'll look at is we'll look at the earliest microscopes, talk about what the elements of good microscopy are. We'll look at light versus electron microscopy very briefly, and we'll talk about differential staining and how we use microscopes to identify different kinds of microbes. So, microbial dimensions. I do not want you to memorize how large or small different types of microbes are. What I do want you to have an idea of is viruses are generally the smallest, and then there are small bacteria, then there are larger bacteria, then yeasts, then protozoa. Um, just and fungi are usually long filamentous strains of cells. So once they get to that size, you can actually see them with the naked eye. So I would rate them. So yeasts are a type of fungi, but they can be present as a single cell. So they're a bit smaller than a total fungal organism. So have an idea of what's small and what's big in the world of microbiology. This just gives you another way to look at that same information, right? So you've got this is a reproductive structure of a bread mold or a fungus. This is a louse here, which we'd consider to be a helminth, right, or a small insect. Those would be the largest of what we would be looking at. Down here at the smallest end, something like a hydrogen atom or an amino acid. Above that, viruses, prions would be um, very small, and we would need an electron microscope to look at those. Um, uh, then on to things like bacteria, which are, or yeasts, which are comparable in size to red blood cells. Some of the protozoa, like Gerardia, would fit into this group as well. We can use a compound light microscope to look at these. And these guys we can see with the naked eye. So magnification. So a clear glass sphere can magnify or make most objects look bigger, right? It comes from a complex interaction between visible light and or light waves and the curve of that lens or the curvature of that glass. So the first microscopes were just really nothing more than a simple magnifying lens that had a few working parts. It might have had like a little screw that acted as a stage. Later, compound microscopes, which are the kind that we use in most college microbiology labs, have a second magnifying lens that we'll look at in a minute. You have an ocular lens and an objective lens. You have a lamp in the base that illuminates the specimen, and you have a condenser that focuses the light on the specimen. So this is what a compound microscope looks like. This is where one of those places where I really feel like asynchronous online learning fails you as opposed to really working in a lab. You'll do lots of virtual labs where you'll do some microscope experiments, but it's not the same as learning how to use one and really get a good feel for that. If you're going into work into a laboratory where you need to use a microscope, you really need that kind of hands-on experiential training. But we'll go through some of the parts here. So this is the ocular up here where your eyes would go, also called the eyepiece. That ocular has a 10x magnification. The body of the microscope is the metal body of the microscope that holds all the rest of it together. You've got an objective lens, actually several down here. You usually have a 4x, a 10x, a 40x, and a 100x objective lens, and we can turn this around in each of those different powers. So to get the total magnification, we would multiply the objective lens, let's say that this is the 4x, times the ocular lens at 10x. That would be a 40x magnification. If we use the 100x oil immersion lens, it would be 100x times 10x, which would be 1,000 times the magnifying of the original size of the sample. This black metal stage is called a mechanical stage. It moves forward, back and forth, and left to right. We have a condenser underneath the stage, which will help to condense the light source and direct it towards the specimen. We have a light source in the base of the microscope that we can dim and make brighter. And then we have focus, the stage adjustment knobs, which move the stage around, again, left to right or back and forth, front to back. And over here are your focus knobs. You'll have a coarse focus and a fine focus. The way that this works is when we make a slide, we always start at the lowest power, at the 4x power, to find the specimen and get it into view and into focus using the coarse focus first and then the fine focus. Then we move up to the next power, which is the 10x, and we should only have to adjust our focus at a minor level there if we had it in focus at the 4x. And then we get it in focus at 10x, and then we can move up to the 40x. Sometimes there's a light intensity control on the body of the microscope. Usually that light intensity, it depends on how you turn on your light down here. You may have a dimmer switch. You may have a light switch that turns it on. That varies quite a bit from microscope to microscope. Let's go back to the clips. Slide clips, you can see these two silver metal clips here on top of the stage. Those open up and you slide your slide in where it secures into those clips. So 3 properties of having a good effective microscope are the magnification, right? How much can you magnify your sample? Resolution, how clear can you see that image? And contrast, which is really light against dark, which helps us again visualize the image. So the amount of contrast you can achieve helps us to visualize the specimen. So that ocular lens, the one closest to your eye, also known as the eyepiece, makes what we call a virtual image. It's what your eye sees. It's a retinal or visual image. The objective lens, the one that we're using that's closest to the specimen, makes the real image. So this is how that works. You have your light source down here. The light passes through your specimen here on the stage. That objective lens is creating the real image of that microscope slide or that specimen. That light continues, it bends here and passes through the ocular where you're making a virtual image that your eye can perceive. Again, magnifying power of the microscope, you calculate it by multiplying the ocular lens, which is always 10x, times the power of the objective. If you have a 10x low power, then it's 10 times the 10 of the ocular. That's 100x magnification. 40x objective times the 10x ocular. It's a 400x total power. 100x oil immersion objective times the 10x ocular is 1,000x magnification. Wavelength on resolution. So we're constantly with that condenser and that aperture trying to adjust our light source and so that it passes through our specimen without a lot of diffraction. So we're trying to make a tight, clean image. And so you can play with your light to make that happen and play with your aperture to reduce the amount of light that passes through your specimen. And what that does is it can change that diffusion, that light diffusion where you have a very blurry image to making a really tight, clean image that you see. Oil immersion lens, I believe you'll get to do a virtual lab on this. We put a drop of oil on your sample before moving the 100 X objective into place, twisting it into place. So that oil has some of the same optical qualities as glass, as long as your oil's clean and in good shape. And it prevents the scattering of light rays or that diffusion of light, so it should increase your numerical aperture and your resolution um, of that microbe or of that sample. Oil immersion, when it's right, should be able to resolve objects that are 0.2 microns apart. So this is nothing more than your objective lens, your 100x objective lens. Here's your slide down here. You've gotten it into focus at the 10x, then the 40x. Then we add a little bit of oil to it, and we will roll that 100x into place. It's actually so close to the slide that you can scrape or crack or damage your 100x lens if we don't have this oil present. So it's doing two things. It's protecting the objective lens, and it's hopefully adding to the resolution of that image. So variations on a light microscope. So we would be using a compound microscope in our microbiology lab. It would be a bright field microscope, meaning that the background on our samples would be light and our samples would appear dark. You can have a reverse of that and have a dark field microscope as well. There's a phase contrast microscope that gives you kind of a three-dimensional image based on, you can kind of see an outline, a three-dimensional image of the sample that you have. There's also an interference microscope. Many people use fluorescence microscopy when we're looking at tissue-specific staining, when we're looking at, like, if you're looking, yeah, if you're looking at different cell types altogether, and what we can use are different fluorescent dyes that are attracted to each of those, some component of each of those different cell types. So you can see here, this is an example of fluorescence microscopy where we've used a GFP dye that is attracted to one kind of tissue type, and a RFP, a red dye, that's attracted to another tissue type. Usually this blue type, is called DAPI stain, and it's attracted to DNA nuclei. So you get this kind of differential fluorescence microscopy, which makes incredible pictures. This is a type of and it uses a UV ultraviolet radiation as the illuminating source, which excites these different dyes and gives you that kind of differential staining. Confocal microscopy can also give you that kind of fluorescence. We can use that with live samples and actually look at how those dyes, as they're attached to different tissues or different proteins, how they're moving around in a living system. You can also use a super resolution light microscope that uses two different lasers. All of these are very expensive. Again, most of what you'll use is a compound bright field microscope in most microbiology labs. But as you move on, if you move on to education, graduate level education, you may be asked to use a lot of fluorescence and confocal microscopy. So preparing specimens, it depends on whether that specimen needs to remain living. We need to look at action in living tissue, or if we're just looking at a microscope, microbiology sample that doesn't need to be alive and moving. We're trying to observe, we're trying to find that microbe, we're trying to identify that microbe, and in some cases, we're trying to look at movement of that microbe. And so and that determines the type of microscopy we're going to use. So, wet mounts are the most common thing that we use in a microbiology lab. Those cells are suspended in some kind of media, usually a broth, or they're suspended in water. Um, and that fluid maintains viability of the cells, and it provides we can get a true assessment of the size, the shape, the motility, and the color of the cells as long as we're not. So that's what a wet mount allows you to do, is allows those microbes to move freely. So we'll talk about a hanging drop mount, which is probably the best way to do that. So a wet mount is nothing more than you have a microscope slide, you put a droplet of your sample on there, and you overlay it with a cover slip. So it's quick, it's easy. It can also damage your larger cells. It dries out really easy, and you can have contamination because as soon as you drop that cover slip, right, it all runs off onto your hands as an observer. So that's one way to look at living cells. Another way is to make this hanging drop mount. So this microscope slide has a little divot in it, a dish, where we can drop a drop of liquid culture, um, microbes in media or in a cell culture, um, where we can drop that into the microscope slide. We cover it with a cover slip, but they've got this little swimming pool down here to swim around in. We use Vaseline to seal up the cover slip, and that gives us a way to really look at movement and a living sample. It does overcome the disadvantage of wet mounts. You don't get any on you as the observer. You're not squishing the cells. It doesn't dry out. So they're very nice to look at living samples. The most common way that we look at samples in a microbiology lab is by using a fixed smear technique, which you will do in the virtual lab. So we take a little bit of water, we spread it on a slide, we use a sterile loop, we take a little bit of our sample, might be from a solid plate, might be from a liquid culture, we mix it in with the water, we allow that slide to dry, and so this is not a living sample. Then we heat fix the slide. Once it's air dried, we heat that slide gently in a flame. It kills all the cells, it secures that specimen to the slide, and it keeps all those cellular components in a natural state with minimal distortion. Some people also use methanol, alcohol, formalin to fix their slides, depending on what they're trying to do. So staining those smears, we're providing contrast, right? And you're using a bright field microscope. You've got a bright white background, you want a dark dye. So it makes inconspicuous features stand out. Those dyes like methylene blue, crystal violet, safranin, I think you'll use a number of different things in your virtual lab. They impart colors to the cells by fixing affixing themselves to those cells through a chemical reaction. So basic or cationic dyes have a positive charge and they're attracted to acidic, negatively charged components on bacterial cell walls. Acidic dyes or anionic dyes have a negative charge and they're repelled by acidic, negatively charged components on bacterial cell walls. A positive stain is a positively charged stain is attracted to negatively charged cell walls. It sticks to the cell and it gives it a color so you can see the cell. A negative charged dye is repelled by those negatively charged cell walls and it produces a black background around the cells. So that's all great, but let's see a picture because that's probably better. Hold on, let's skip a couple. So I wanted to show you a negative. Well, we're going to get there in a minute. I apologize. So what these negative stains look like is the background will be dark and the cells will be kind of translucent and light colored. With a positive stain, the background will be light and the cells that you're looking at will have the color of the stain. All right. Oh, there it is, sorry. So negative stains, this is what it looks like here on the right. You've made a dark background, and you can see the cells in a lighter color on that black background. We really don't use this very often. This is an example of positive staining with the bright background and the dark cells on top. Um, so obviously with the positive staining, the background's not stained. Here with the negative staining, it is. Positive staining, these are all examples of really common dyes that we use, like crystal violet, methylene blue, malachite green. These are all dyes that we use for positive staining. For negative staining, you have these acidic dyes. One is called nigrosin, one is called India ink. Um, so subtypes of stains, you can have very specific stains. You will get to do many of these in the virtual labs. Differential stains, gram stains that help us determine gram positive from gram negative bacteria. Acid fast staining, which help us determine acid fast bacteria. So let's look at pictures of some of those kinds of differential stains, because you'll do all of these. So this is a simple stain. You've done nothing more than take a sample, heat fix it to a slide, then stain it. I think this one is methylene blue. Oh, nope, sorry, crystal violet of E. coli here. The E. coli cells are in blue purple on a white background. Here is a methylene blue of trichomonas, where the trichomonas is blue and the background is white. It's uncomplicated, takes a single dye, is very effective. The differential stains that we'll look at usually use two differently colored stains so that you can tell two types of cells or two types of cell structures or cell parts apart from one another. So let's look at that for a minute. Gram staining is something that we use all the time. It's very important in identification. If you have, if you're looking at an environmental sample that you've isolated, and it could be one of 100,000 different kinds of bacteria, right? To narrow down your search, a really common thing to do would be to determine, is it gram negative or gram positive? Because once you've done that, you've excluded, the other half. So gram staining will do that. Gram positive bacteria, uh, crystal violet stain embeds itself in the cell wall of gram positive bacteria, making them look purple. That crystal violet does not embed itself in gram-negative bacteria, because they have a single layer of peptidoglycan, so there's not much there to catch the crystal violet. So once we restain with safranin, those bacteria look pink. So the gram-positive bacteria have multiple layers of peptidoglycan in their cell wall, and it traps that crystal violet, making them look purple. And you can see here we have a mixed culture, right, where we have gram-positive bacteria in purple and gram-negative bacteria in pink. Acid-fast staining. So there are certain kinds of bacteria that are acid-fast, certain that are not. So the agents of TB, tuberculosis, and leprosy are acid-fast. And this allows us to look at them in a bigger culture. So the red cells are acid fast, the blue ones are non-acid fast. You'll do this in the virtual lab. Endospores, important. These resting structures that certain kinds of bacteria like Bacillus and Clostridium can make when the going gets tough. So if pH is bad, temperature is bad, nutrients are low, these bacteria can make endospores. And endospores are incredibly resistant to temperature, to antibiotics. They are a resting structure that can really wait for a very long time and then reemerge again. As such, they are a problem in food preservation. So we talk about, and we'll talk more about this, I think, when we get to bacteria later. But you can stain for capsules. Now, in our lab, the malachite green actually stains the spore structures, and the pink stain that we use will stain the cells. Here it is reversed. In this particular case, the endospores are in red and the vegetative cells are in blue. But this is how you would look at this and say, well, yes, this bacteria can make endospores, which means it's either part of the genera Bacillus or Clostridium. Differential stains. Capsule stain is a capsule is an unstructured protective layer that you see sometimes on some bacteria and some fungi. We look at it by using negative staining so that in the ink or Nigerson stain, you can see if a bacteria has a capsule, and only certain bacteria do, like Cryptococcus, which is pathogenic. Um, this is a Cryptococcus slide. So if they have a capsule, it will kind of glow on that negative background. Flagella are these appendages that bacteria use to move around. This is Proteus vulgaris. They used, you can use flagellar staining in order to look at the types of flagella you have. That the stain, so flagella are made up of their own protein, so you can have stains that are particular for flagella, and they'll stick to the flagella so that, yeah, you can see them. And this gives you a chance to go back and look at some of what we've gone over with microscopes. Again, not a comprehensive review. Always take good notes with your slides. Go back and review those notes before you attempt the weekly quiz.